गुड मॉर्निंग सभी का इस वीडियो लेक्चर में स्वागत। एमएससी फाइनल बॉटनी पेपर 5 के इस वीडियो में जिस टॉपिक पर चर्चा करेंगे वह है एपोमिक्सिस। द नॉर्मल सेक्सुअल साइकिल एपिमिक्सिस इन्वॉल्व टू इम्पोर्टेंट प्रोसेस। फर्स्ट इज meiosis, second is fertilization. In sexual cycle, diploid generation alternates with a haploid generation. In some plants, meiosis and syngamy are interrupted and still have a viable embryo is formed within the confines of the second seed coat. Such asexual seeds which produce progeny identical to the female plant are called apomictic seeds, and phenomena is known as apomixis. Apomixis meant act of mixing or mingling. Apomixis has been reported in more than 300 species belonging 35 families. It is most common in families like Asteraceae, Poaceae, Rosaceae and Rutaceae. More common in polyploids than diploids. There are two main categories of apomixis. First is vegetative reproduction or second is agamospermy. Two different types of agamospermy are recognized based on whether the embryo develops from a cell of the unreduced female gametophyte or directly from diploid sporatic cells of the ovule. In gametophytic epimexis, an unreduced embryo sac develops from the microspore mother cells by circumvention of meiosis, diplospory, or directly from a cell in the nucellus called apospory. Vegetative reproduction. Some form of vegetative reproduction is generally found associated with the seminiferous plants reproducing by means of normal seeds. However, only those plants are regarded as apomictic where vegetative reproduction has been replaced the sexual method of completely or essentially so. It includes by means of bulbs, runners, suckers, and so on. Gustafson 1946. There are three types of vegetative reproduction in higher plants. First, the propagules are formed outside the floral regions despite the occurrence of functional sex organs. No fertilization or seed setting takes place. Second, the propagules are formed outside the floral region and the plants are sexually sterile. Third, the propagules are formed on the floral branches either in addition to or in place of the flowers, the phenomena called viviparity. Vegetative viviparity is quite common in grasses. It can also be induced artificially. Agamospermy. Adventive embryony. In this type, the gametophytic generation is completely eliminated. In this respect, it is close to vegetative propagation but differs from it essentially in two ways. It has retained the seed habit. Second, one or more diploid sporophytic cells undergoes the stages of embryogeny and grow into mature embryos inside the normally developed sexual embryo sac. Adventive embryony usually leads to the formation of more than one embryo in a seed. Agamospermy. Gametophytic apomixis. Diplospory. In this type, the microspore mother cell differentiates as in sexual ovules, but it does not enter meiosis. It produces diploid embryo sac. Example, Antennaria alpina. Four types of diplosporic embryo sac development have been described. First is Terexcum type. Second, Icaris type. Third, Antennaria type. And fourth, Allium type. The chromosome number in microspore mother cell is doubled by endomitosis and the ensuing meiosis. Results in a diet of unfertilized cell. Two subsequent mitoses in the chalazal diet cell results in an 8 nucleate embryo sac. Allium nutans and allium Odorum so allium type of diplospory. Apospory. Apospory is by far the most common mechanism of Epimexis in high plants. It was first reported by Rosenberg 1907 in Hyeraceum species. Obligate nuclear apospory with pseudogamy has been observed in 7 species of Pennisetum. In aposporous plants, a normal microspore mother cell differentiates in the same manner as in sexual ovules and begins to enter meiosis, which may or may not complete both divisions to form a linear tetrad. At some stage during this period, one or more adjacent nuclear cells become meristematic and appear like microspore mother cells. These cells undergo mitotic division to form unreduced embryo sacs. In obligate apospory, the sexual microspore mother cells or it products abort and the apospora embryo sac occupies the entire area of the ovule. The timing of the degeneration of sexual embryo sacs depend on the time of initiation of the aposporous embryo sac. The sooner an aposporous initial is induced earlier, the degeneration of the developing megaspore occurs. Occasionally, aposporous and sexual embryo sacs may coexist. Apospory is mainly of two types. Hyeraceum type in this MMC undergoes the usual meiotic division and forms a tetrad. At this stage, a nuclear cell at the serosal end of the tetrad becomes activated and starts developing into an aposporous unreduced embryo sac. Second, Penicum type. In Penicum and some other grasses, the aposporous embryo sac is 4 nucleate organized into a 3-celled egg apparatus and a polar nucleus. Antipodals are absent. Embryo and endosperm development. Diplospory as well as apospory produce diploid embryo sacs. To complete the apomictic cycle without altering the chromosome number of the sporophytic generation, the diploid egg must develop into embryo without the participation of the male nucleus. Formation of embryo from an unfertilized egg is called parthenogenesis. Although the embryo development in apomictic occurs without the union of male and female gametes, pollination is often necessary for full development of the embryo and endosperm. Based on the timings of embryo and endosperm development, the apomictics can be divided into 3 categories. First, in some apomicts, embryo and endosperm development are autonomous, not requiring a pollination stimulus. Most apomictic members of the Astraceae show autonomous endosperm development. Second, precocious development of embryo before pollination has also been reported in Parthenium and Poa species. But in these cases, pollination is an absolute requirement for endosperm development called pseudogamy. In another category, third, of pseudogamous apomictis, to which belong Tripascum dactyloides, the embryo starts developing after endosperm development, suggesting that here the gene S involved in the regulation of parthenogenesis is are expressed only after the polar nuclei are fertilized. In pseudogamous apomicts where embryo development occurs before pollination, category second, by the time of sperm cells reach the female gametophyte, egg cell fertilization is no longer possible, but fertilization of polar nuclei for endosperm development still takes place. The hypothesis is based on the observation that apomictic embryo sacs, which bypasses 2 processes in normal embryo sac development, viz. 2 meiotic division and megaspore degeneration phase, accuse fall development in shorter time than the sexual embryo sacs, resulting in a gap between receptivity of unreduced egg cell and the general floral development, that is pollen production stigma receptivity. This gap prevents egg fertilization, but endosperm development can still be induced for normal seed development. Practical importance. Apomixis was once regarded as an evolutionary dead end because the meiotic segregation and recombination on the female side are eliminated. However, now it is being regarded as a potential powerful genetic factor for use in crop improvement. Apomictically produced plants are genetically identical to the female plant. Therefore, if Apomixis can be introduced in crop plants, it would provide an inexpensive way to perpetuate a given genotype, preserving such characters as heterosis through successive seed generation. Apomixis would also simplify commercial hybrid seed production. However, Apomixis is not very common in the major crop plants. Inspired by the potential benefits of apomictic reproduction, plant breeders are making serious attempts to introduce this trait into crop plants through ecotypic selection, sexual hybridization with an apomictic relative, induced mutagenesis, and genetic information. Development and release of apomictic cultivars of Poa pratense and Cenchrus ciliaris have demonstrated the potential of Epimexis in breeding program. Adventive embryony, well known in citrus, is being used to production of uniform root stock and virus-free सियोन मटेरियल. इस प्रकार इस वीडियो के अंदर अपने एपोमिक्सिस के बारे में डिटेल स्टडी किया, जिसमें व्हाट इज एपोमिक्सिस, देन इट्स टाइप और देन इट्स इम्पोर्टेंस. सभी का शुक्रिया, धन्यवाद.